Hello, I am Amanda Wacker.
Hola, I am Lennice Castro.
สวัสดีค่ะ I'm Asama Lekbua.
And we are the Triplicates. Welcome to our podcast. We are three first-generation women of color currently in our first year of our PhD at the University of California, San Diego.
So, what are we talking about today?
I think we should talk about our experience so far. We are almost at the end of the first quarter of our PhD, first year, kind of a big milestone. Did not expect to be here, I think. Kind of wild. So we can start from the beginning. Should we start with bootcamp?
Yeah, I don't know. That's like not. I don't know. Is that normal for every grad school?
I don't know.
We start just with that. Explain a little bit.
Yeah, because technically we earn credits from bootcamp, so that's like part of our curriculum.
Oh, we do. I forgot about that.
But it's before the first quarter actually starts, right?
I know, but it's like included in our
And it's
first
required.
quarter credits.
Yeah.
It's like 2 credits.
I'm not sure how that works. Like,
I don't
did we start in the summer technically?
Yeah.
Wow, is this our second quarter then?
I guess technically.
Wow.
So we did a boot camp.
So it's not like military boot camp.
No, it is not.
Everything was on Zoom.
Yes.
5 hours of Zoom every day.
It was a lot. It was a lot of Zoom in.
Yeah, so it's basically, for those that don't know what a science boot camp would be, it's kind of just taking a dive into science and just shifting your brain to like start thinking about scientific research again. And after a long break, maybe people are just got out of school and so they haven't really thought about science, or they're coming from a non-academic lab and they're coming back into this scientific research at a university type thing. And so boot camp's supposed to serve as like you getting used to that before you start your rotations and stuff, and normal classes. But on that,
I feel like it was like a trial run, or like it's like, you know, when you like join a job and it's like, okay, you're gonna like just train for the first 2 weeks. Like that's what it felt like. It's like, okay, you guys are gonna read papers and practice presenting and listen to us talk about things you guys should know already, but we're not gonna be too strict about it.
Yeah, I think it was also, we haven't gotten to like interact with our cohort that much due to the fact that we are living in a pandemic.
Yep, still here.
Yep. Surviving and thriving.
Are we?
Well, some weeks we are, some weeks we
Some weeks are better than others. Well, I think it's week 10, not counting boot camp, and boot camp was 4 weeks, so we've been here.
Are we in week 10? I thought we were week 9. Obviously, I don't keep count.
This will come out week 10 then.
Oh, yeah.
Yeah.
Yeah, you're right.
Boot camp is not all about like taking classes and being on Zoom. I think my favorite part, well, I had 2 favorite parts. One was when a professor came and taught us how to write grants. I thought that was really helpful, and I ended up working on a lot of grants this fall, and I thought that was a good way to sort of like introduce what that process is like, because a lot of grad students and also later on in your academic career, like you would have to do that. And my other favorite part was when we got to do a micro rotation.
I forgot we did micro rotations.
Right. And in a lab that I personally would not even think about, because I love bacteria and I love molecular biology. And this lab that I got to rotate in was a worm lab. So I got to learn about C. elegans.
C. elegans.
Yeah. What were your micro rotations like?
So I also love bacteria like Asama, and that's mostly what I did my work in. But the lab that I worked in for my micro rotation is actually in yeast.
What?
And we did study like the division of ER, aka the endoplasmic reticulum.
Wait, whose lab were you in?
Mahoniwa's.
Okay.
So I'm currently rotating in a lab right next door, so I see them here and there. But it was good. It was short.
It was 3 days.
Yes. So we didn't get much done.
But it was the first time we were able to be on campus.
It was also the first time I had touched a pipette in like 8 months.
Oh,
It was nice.
Yeah.
That's real.
I liked it.
I was in a butterfly lab.
Oh, what?
Yeah, it was. I don't think I would have. They do like vision and development of visual systems in insects. And I don't know. I don't think I would have. I don't know. I like you guys said, I'm in bacteria, I guess, more. So I don't know if I would have been a little out of my element to be like, let me just go work in this butterfly lab. But it was really cool. We got to like inject eggs with CRISPR.
What?
And I was able to pin like a butterfly like they do in museums, which was really cool.
Yeah.
I was very scared of touching bugs. So we did that, you know.
Did you get to take them home?
I think he said we could take them home if we wanted, but like I never went back to get mine. Maybe I should have. But like, where would I put it?
I feel like that's a cute little decoration.
Maybe
somewhere.
Yeah. You can give them to me.
Yeah, I would. That's like a very aesthetic Lennice vibe. You know? I don't know.
Dead insects on your wall.
Stop.
Okay. Anyway.
So yeah, I liked that, but I guess it was the first time that we were exposed to people in our cohort. Like we could actually interact with them, even though it was over Zoom / my micro rotation. I did it with one other person, so I was able to get to know someone and finally like interact with humans again.
Yeah.
So it was nice to see people from like all kinds of different backgrounds, like both as people and like science wise.
Yeah.
And see how they think and what we kind of bring to the table, like offering different perspectives. And I think the fact that we collaborated so much during our micro rotation and boot camp speaks to like the greater goal of like the division of biosciences. Like it's a very collaborative division, so it was nice to kind of get that like just from the jump.
Yeah, I wasn't ready to be doing group projects in graduate school.
I heard it never ends, even when you become a professor.
Yeah, well, now that you mentioned that, I guess that does make sense that you're always going to be like collaborating with people. But I was like, I wasn't. Yeah. We did, I think, was it 2 group presentations?
Yeah,
at least 2.
Yeah, Amanda was in one of mine, and I thought she was really cool.
Oh,
that was before we became friends.
Yeah,
I was like very shy.
I
I still am very shy. If you can't tell.
You're doing great.
Thank you.
Okay, so we did boot camp, we did micro rotations, and then quarter technically started October.
Yes.
5th, I think. But our first rotation started maybe October 1st.
Yeah.
Yeah.
So our rotations, how they work at UCSD, are 6 weeks in a lab, and we get to choose where we want to go. It's kind of a short time frame of trying to get into a lab. From boot camp, they release the list of people that are accepting students, and then you are kind of just in a race with the other students trying to get into their first rotation.
I remember being so stressed out because I wasn't sure if I should contact one person at a time and then move on, or should I email like 6 people at once. And funny story, Lennice and I have very similar interests. So we had our eye on the same lab, and then I emailed a little later, but then I ended up not booking that lab for my first one, which is fine. Like I booked that lab later on, but then
I'm waiting for the shade.
It's coming. It's coming.
And then I was just going around the cohort like, hey, who's doing what? And Lennice is like, oh, this lab.
You're like, oh,
it was you.
It's a you. It's fine.
But it's okay. You're rotating with the Dutton lab soon.
Yeah, and we'll be there at the same time.
Yeah, and I get to be there with Amanda, so I don't know who's
So who
jealous
won,
enough.
actually? It's Asama.
Excuse me? I will be across the way. We're going to get nothing done.
Don't say that. They listen to our
Oh,
podcast.
yeah. Okay, well.
All of them.
We will be working so hard.
Very hard. Independently collaborating.
We
sometimes
will.
only.
Yeah, but should we take a step back and explain exactly, like, what a rotation is? Or, like, what a program having a rotation-based program means?
Sure.
I don't know. Should I explain? Okay, I'll go.
Yeah.
Some places have what we call direct admit, and some places have rotations. And usually when you have a direct admit, it means that you're applying to a specific lab at the university within whatever department or division you're going to be in. But rotations, a rotation program like the one at UC San Diego, is where you kind of test the waters in a lab and you're able to work there for a set amount of weeks. Like Amanda said, here at UCSD, it's about 6 weeks. And you're able to get to know the people in the lab, get to know the science, because you get a project or you think of a project to do. It's like a vibe check, to be honest.
It really is a vibe check. There's no better word for it.
Yeah.
It's really just a vibe check.
Because when you think about it, like at the university level, like any lab, I hope it's like scientifically rigorous enough that whatever research you do is impactful and it's important and it's cool. But the vibe check is really for you to see if this is something that you are personally interested in. You can do this in a longer term, or like seeing if this is like your people, like in the lab, and like how your working relationship or like friendship with the people are like. So I often got this advice at the beginning of my rotation that if you think the science is cool, like the lab down the hall, the science is also cool. Like every lab, the science is cool, but you should focus on finding your people.
And that's also what's great about being in an umbrella program. So an umbrella program just means you're not limited as to what science you do.
I mean, there is a limitation. So, like,
that's true.
you could go into a specific neuroscience program. Like, I think there's a neuroscience program specifically
There
here.
is, yeah.
But under the Division of Biological Sciences, there's some labs that are in neuroscience that you can rotate in, but you can also rotate in things that are doing developmental biology or cell molecular stuff. So you're not limited in that. But if I wanted to go rotate in chemistry, I have to wait until the summer to do those rotations.
Yeah.
That's true. You kind of, if you're thinking you might not want to stay in the field that you've been in the rest of your academic career, in an umbrella program, it gives you that freedom to kind of test the waters in like departments or fields that you haven't been exposed to, which is something I really liked because like I previously said, I've been in bacteria/bacteriology my whole academic career. But I kind of, I don't know, I want to do like a little immunology or virology stuff. I think that's kind of cool. Yeah, so a lot of my rotations are including that. And yeah, me and Asama have similar rotations, so her rotations are also including that.
I think we also have two overlapping. I have two overlapping with you. And what I do like about the umbrella program is, like you were saying, switching. Like, I come from a mouse background. I've only ever done mouse work, really. So to come and be like, okay, actually, I don't want to do mouse work anymore. I want to switch to something else. Like, to be able to test out, okay, which... Do I want to do yeast? Do I want to do cyanobacteria? Do I want to do fungi? Like, to be able to test that all out is nice. And not be, like, as stressed about, like, okay, am I picking the correct model system?
Yeah. And you learn so many new skills. I don't know. I feel like I've picked up a lot of, like... skills and soft skills during rotations, even though we've only done 2. Like, it's just, I don't know, I've learned a lot,
especially soft skills during COVID.
Yes,
it's hard.
Yes,
COVID is hard.
Amanda has helped me improve my emailing.
Oh, girl, I love an email.
I love it. She loves an email. Hear
is,
that, everyone? triplicatespod@gmail.com.
Okay, yes, plug us. You can email us. But, like, I think the thing with emails is, like, I don't know, I like talking to people, and it's, like, the most direct way to talk to people, and you just can be like, hey, hope you're doing well. These are all the things that I need to be addressed within the next 2 to 4 days, maybe. And it's like, I don't know. I'm good at emailing. I'm really bad at doing subject lines, though. Like, I would just be, like, very, very blunt, like, whatever. Like, very just, I don't know. I wish I had more finesse
Get straight
with
to
that
the
one.
point.
Yeah, exactly. But, like, that's the point of a subject headline, I guess.
Although, during my time in industry, we were coached on how to email each other if we really need a response. So, like, sometimes in the subject line, we're advised to say, please respond by blah, 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 in a bracket, and then say your subject line.
Wow.
Because if you're emailing someone who has a million emails, like, that's how they can prioritize just based on glancing at their inbox.
Maybe I should be doing that now.
I've never heard anyone say that before.
I've never thought of that.
See, this is, okay, so the thing is, me and Lennice came straight out of undergrad.
Yes.
And Asama, how many years did you spend in industry?
3 years consecutively, like, after undergrad, but I've interned in another biotech for like 6 months before. So
wow.
yeah, I spent a lot of time in biotech, and their soft skills are different. Like in many ways, people in academia would say that they're more cutthroat or more direct, but in other ways, I think they're just forced to be more efficient.
Right.
Just by that email example, because if they don't see the subject line and they don't respond in time, then like, I don't know what else to tell you. I already told you when I need this.
Right.
Yeah.
Interesting.
Maybe I'll use that. Because recently I've been asking for letters of rec.
Have they not replied?
Well, I don't want to out people, but one of the PIs I asked for a letter of rec. It's not out of character. He requires many reminders, which is fine. Busy person. But maybe I should do that at the deadline of the fellowship. This is when I need this. And that'll get their attention. I've just learned to, like, if he emails me, email him right back, right then and there.
Wow.
To get a response, kind of thing.
Okay, I feel like we diverged a little bit. What were we talking
about? We were talking about rotations.
Rotations. So we mentioned we did 2 rotations. So we're in the middle of our second one. Do we want to talk about our first one a little bit? It was 6 weeks. Do we feel like we learned something? Do we feel like we got what we were supposed to get out of it, which was the vibe check?
For sure.
Yeah.
I did, yeah. I think I was very happy to rotate in the lab that I did, which was the Dutton lab. I always thought the science was super cool. They're the cheese lab here at UCSD. Everyone knows them by that. And that's because they study microbial communities and formation in cheese. They were so welcoming. I felt like just like warmly welcomed, and I felt like I could be myself and just chit chat to people whenever there was another person in lab with me, because we were working under restrictions due to
it's a luxury
COVID.
These days to see another human
it
being.
Really is. It
in
really
the lab.
is. What's nice though in lab, we're almost always in we are in full PPE.
Yeah.
If you're not like sitting on a desk and doing computer stuff. But, yeah. What about you guys? Did you?
Yeah, I think my first lab was a really great choice. I felt really welcomed, and it was supportive, and the other graduate students were really trying to be super friendly to me, which I thought was good. And I feel like that was a good transition from going into grad school to being like, okay, I'm not sure what's going on, and then to be like, okay, I'm safe, I'm comfortable, I can do this. And then that really helped me, I think.
What about you, Asama?
Yeah, I think like talking about the transition between like where you were and your first rotation was something that I also experienced during my rotation, because like I said before, I worked in industry for 3 years before going into grad school. So I did not want someone to like hold my hands and like micromanage and watch me pipette and doing all of that. So for my first rotation in a Doherty's lab, I thought that I was like my own project manager. Like if I needed something, I would just talk to the professor and then we would just like figure it out. I also liked how everyone had autonomy and were in charge of their own project. So I thought that piece was really cool. So the vibe check for me were like everyone's independent and everyone's great at communicating and people are really nice. So I had a great time.
That's interesting. I didn't know what the perspective would be coming in from industry, but like it makes sense that you would want to be like not left alone, but be like, I know what I'm doing. I want you there. Like if I need you, I'll come to you. But like, let me try to do this by myself first.
Yeah, and this is something also I learned in industry is to try to give feedback, even though it's hard to tell your boss not to micromanage you. But yeah, we learn, and I always ask to sort of like set up dedicated meetings so like we can ask questions, we can talk about things, but when I'm in the lab I want to try to do everything, figure it out on my own, because that really helps me like concentrate, but also like try to solve problem on my own before like immediately like trying to find someone.
Yeah, it's funny that you mention like hand holding, because I remember coming in, I was like, I know I can do science, but here's my hand, please hold.
You know, cover it. Like, you know when you're like teaching a kid on a tricycle, you're like, I'm here, I'm right
here. Yeah. But my first rotation, I feel like I really kind of took the wheels, and I was, due to the restrictions, I was in lab alone a lot, and did like a new type of experiment that I hadn't done before on my own.
Mhm.
And we had some trips, we had some falls, but listen,
Not physical ones,
Not
right?
physical ones.
Okay. Just to be clear.
but kind of being thrown in the water and just doing what I need to do and trusting myself,
Mhm.
Yeah.
knowing that I can do it. It really just reinstilled the confidence into me that like I can do science. Like
Of course you can.
I don't know why I'd be doubting myself, but I shouldn't because I can do it.
I mean, you're here.
I
You got
know.
in.
Yeah.
We're all here. We
Yeah.
all got in.
Yeah. But that's how I feel about I was doing microscope
Mm-hmm.
stuff by myself.
Mm-hmm.
And the first time they did the training, so I was trained by someone from the core facilities.
Mm-hmm.
And the next time they're like, I'll help you set up and then you can go.
Mm-hmm.
And then the third time I went, I was having some sort of issue. It wouldn't recognize the thing. I was there for 30 minutes trying to figure everything out. And then I finally sent the guy the email and I was like, hey, I've been trying to do I did this, and that.
Mm-hmm.
And then between me sending the email and me looking at it again and be like, okay, let's try this one thing one last time. I did it already, but let's try it one more time. And then when I did that, it worked. And I was like, okay, actually, I'm good. We're chilling. And after that, I was like, okay, you know what? I can do anything. It doesn't matter. I can do anything. It just takes maybe 35 minutes.
Yeah.
But you know what? You make it work.
Yeah, definitely those first few weeks of being in lab, I was like, I'm slow. It's
Yeah.
gonna take me a little while to get through these things, but
Mhm.
I hadn't been in lab in so long, and
Right.
it's my first time on said project, right?
Mhm.
So I think it was a humbling, but also confidence boost experience that I had that first rotation.
That's nice. And how do you
Yeah.
feel in your second rotation?
I like it. So actually, in this second rotation, I shifted into virology, and something new was I had never touched tissue culture or cell lines before. I'd only worked in bacteria. So
What's
Yes.
tissue culture?
oh gosh. Okay, so tissue culture. Oh, I don't know if I can.
Tissue.
Oh, I guess they call it tissue culture, but it's mostly working with cell lines.
culture.
Yeah, cell culture.
I see. Okay.
I don't know. They always say TC, which is tissue culture room.
Yeah.
I don't know. That's
I
why
thought they were
called
saying
it.
the cell culture. we said TC room too, but I thought they were saying
Is it the cell?
I don't know.
No, I think it's
'Cause of-
tissue culture.
I think it's
Right?
tissue
See,
culture
See, this
too.
is why I asked because, I don't know.
Okay.
And a lot of
Yeah, I
people
just-
listening might not
Yeah.
know.
We didn't do tissue, we just did cells.
Yeah,
so I
yeah,
just was like, oh,
same.
it must be the cell, but okay.
Yeah. Well.
Like ATM machine, they say the machine twice. The M is for the machine.
Oh,
Right? So I was like,
Shocked.
maybe it's one of those things.
maybe. Anyways, nomenclature. But I'm working with a cell line. So what it is kind of being a mom. I am a mother.
No trauma.
No drama. Maintaining.
This is
cells.
a TikTok reference for all of you not a Gen Z, like everyone else.
So kind of like bacteria, human cell or mammalian cells are similar in that they need nutrients and different things in media, so liquid culture, to stay alive. In bacteria, what you would do is you would back culture, or what was the word you used the other day? Asama?
Subculture.
Subculture.
What is subculture?
Subculture is taking an aliquot and restarting, diluting the current bacteria.
What's an aliquot?
Words.
Yeah, it involves aliquoting, but I don't know why. That's just how
I know, I'm just saying.
People
Like,
train
Like, what is it? What is
some
it?
people don't, I didn't know what an aliquot was till I got into a research
Fair.
lab. And they're like, oh, just take an aliquot. And I was like, how much is that?
it's really cute when I use the word aliquot with non-biologists or even scientists. They would say aliquot, and I'm like, okay, it's like aliquot, but it's like aliquot.
Aliquot.
Yeah.
Anyways, because they're thinking allocate.
Oh.
Yeah.
Is that not the root?
I don't know. You are asking me English terms, ma'am. I am
Listeners,
in
tell us.
STEM.
We know you have
Sorry.
PhDs.
Oh yeah, my English PhDs, bachelors. Please educate us. No, but what is an aliquot, definition wise, for our listeners?
Yeah, you have her over here defining cell culture.
Oh my gosh.
Tissue
Okay.
culture. Here we
Well,
go.
I think to me, aliquot is when you take a subset, a smaller amount of something, and then you distribute it into something else, or you
So you're,
allocate
just taking,
it.
taking
Yeah.
a little portion
That was a
Yeah.
good definition.
and setting it aside. I mean, this is for later.
Yeah.
Yeah. So with bacteria, you do what she calls subculture, I call back dilution. Either way, similar things. You're taking a subset of your liquid culture and putting it into new media so that your cells are happy and growing and have those nutrients to grow. You do something similar with mammalian cells in that you exchange an aliquot of them into fresh media.
Wait, pause.
Okay.
Why couldn't you just change the media?
Because, so, the media, what do you mean just change the media?
Like,
Take out the
you
liquid.
just take out the liquid and put new liquid in. Why are we
You could do that.
moving them?
Some people do that.
What? I guess you take it out. I don't know.
Oh, maybe your cells just aren't adhering. Like, the only time I've done cell culture, they were adherent cells.
Mine
And they
are
were-
adhere. Yeah.
Okay, so why are you...
Maybe they
What?
ran out of room.
I don't know.
Oh,
well,
okay,
we
that's-
trypsinite. Yeah, we. Yes.
So
Okay,
you mean
that's
they
another
need more
thing.
room
You're
to
right.
grow.
Yes.
Okay.
I solved something.
Yes, you did. So, yeah, these cells like to be at certain densities, which we also call a percentage of confluency. So we're splitting you're splitting the cells because
They need more room.
They need more room.
Okay.
So
They got
you
too big.
yeah, too much.
Okay. That
We don't
makes
want
sense.
them to die.
We want
Once
them
again.
to social distance. LOL! Okay, I hope the microphone captured that.
Asama is so funny.
She is.
Okay, so that's what you're doing in your second rotation.
Yep.
Asama, what are you
Ooh,
doing
that
in your
sounds-
second rotation?
Oh my gosh. So my second rotation. So for my first rotation, I worked in a host virus evolution lab, and I was looking at bacterial virus, which is really cool as well. But for my second one, I'm in an ecology, behavior, and evolution lab, EBE for short, and we're trying to set up a system that studies predator-prey relationship between these little, like, small zooplanktons in freshwater lakes and their microbiome and the type of diet or, like, algae that they eat. So it's at the very beginning of the project. So my work so far has been trying to culture these zooplanktons called rotifers. And coming from bacterial culturing background, I was just like bright-eyed, bushy-tailed. I was like, I can do it. Turns out I haven't been able to do it, but I'm trying.
That's okay.
So that's another real thing about science, is that not everyone get everything on the first try. But I'm really glad to have the type of mentorship and the type of support from my lab, because they just look at it and say, okay, you're trying this, you're trying that, that's great. And like, I get it. If it doesn't work, like, we'll try again. So even though I'm not just like, knocking it out of the park like the first time around, I'm really glad to be able to like retry and think things through with my lab.
Mm-hmm.
Yeah, so go ecology.
What about you, Amanda?
So I am working in a cyanobacteria lab, which is super cool. They're little green guys, and they're responsible for the Great Oxygenation Event, which turned the Earth's atmosphere a bajillion years ago. I don't know how many million years ago it was. Technically. Yeah. I don't want to lie on the podcast. But whenever it changed our atmosphere from mostly like CO2 into mostly oxygen, and that is what allowed plants to grow and animals to be animals and literally everything to happen after that.
That's a big deal.
It is a big deal. And there's so many of them, which is super cool. And so the person I'm working under, they're looking at how cyanobacteria would behave if they were on the ISS. So instead of like a 24-hour
Like
day,
the space place?
yes, the International Space
Okay.
Station. Yes. So instead of like a 24-hour day where you have like 12 hours day, 12 hours night, it like is a 90-minute cycle. So 45 minutes light, 45 minutes dark. So you have 16 days and 24 hours, if my math is right, which it might not be, but right. So like that would mess if you have like a regular rhythm in your cell, like that's going to mess.
That would mess me up for sure.
Yeah, I can't. 45 naps are going to be tough with that one. So we're looking at like what kind of gene expression changes happen when you switch their day-night cycle into squishing it into 90 minutes, basically.
So the reason why you're studying cyanobacteria is because they're very well studied, because they've been around forever. Like, what makes them extra special? Why can't we
Yeah.
just observe that in like any bacteria?
Well, people are doing it with a bunch of bacteria, but I think what's
Okay.
cool about cyanobacteria is like they have a really well-studied circadian rhythm.
Oh, okay.
And like it's a big circadian rhythm lab that I'm working in. So the ISS thing is like a side project that we fell into. Like they don't usually do space stuff, but the postdoc I'm working under got a NASA fellowship to do that. So that's why we are into that. Yeah. Super cool stuff.
Wow.
Yeah. I like low-key stalked him before I came here, which is so I had
It's called research.
Yes, it technically is called research. But I had a NASA internship. Oh man, if he hears this, I had a NASA internship this summer, and like my goal was like, okay, let's see how I can continue doing like NASA affiliated research, and like see what else I can work on, and see how my skills from maybe a mouse lab can apply to things I want to do in the future.
For sure. You hear that, NASA?
Please hire me. But I looked at everyone who won a grant in the last like 5 years from NASA, because it's all public, because it's like government funded. So I was looking at like, okay, what schools are they from, like how long is the funding term, all that stuff. And then I found him, and like there's some other people at UCSD doing some stuff, but I found him, and then I found his LinkedIn, because that's the next place you look. And he does like some stuff that's like science communication, and I'm super into science communication. I was like, wait, this might actually be really cool. So I was like super excited to actually like plan it out and be like, okay, this is like what I want to do, what I want to work with. And they were like, yeah, come in. I was like, okay. Cool.
I love that.
Exciting.
Yeah.
Did I say other than tissue culture, what my project was?
No.
Oh, okay. Well, just to say it in like a couple words, is that I was just focusing on this arms race, or this evolutionary arms race between host and viruses. And specifically, I was just looking at viral proteases.
You're saying a lot of words.
I know, I am saying a lot of science words. Protease.
Do they cut proteins?
They do. So a lot of viruses have their own proteases, because they transcribe a big protein, but they can't use it unless they chop it up so it can actually be used to like do virus life cycle stuff.
Wait, so I have a question.
Yes.
When they translate the one big protein, is it linearized, or has it started the like, how proteins fold in on themselves? Like, are they already folded and they get cut where they're supposed to cut, or it's like one big
I think
noodle?
From what I've learned, it's kind of a one big noodle, and then there's like specific restriction.
Oh, and then when it gets cut, they like fold.
Yeah.
Interesting.
Because then they're going to go off and do different things, because some of them are like non-structural proteins, some of them are structural proteins. I figured if they're once again, don't quote me, because if they're folded, I feel like it'd be harder for things to happen, right? Like for these proteases to get in there. But there might be some structural things that like decide where things are going to be cut. So actually maybe there is some foldage happening,
but is that a folding, or is that like a sequence situation?
Well, sometimes you have like secondary structures that can determine.
So it's already started to fold if there's a secondary structure.
So are you?
But you can't say like all of them are, because another thing, like a lot of secondary structures for these proteins, I feel like aren't known or haven't been studied. If anyone wants to study that,
I see a gap.
We love a gap in knowledge. You know what I call that? A grant.
You hear that, NASA? Wait, so are you trying to figure out where these proteases cut, or what's your?
I'm trying to figure out. Oh, I don't know how deep into the science we can get, right?
Break it down. You can do this.
Well, it's because like science-wise, like proprietary information.
Oh, right. Be vague.
Right?
Can you be vague?
Can I be vague? Okay. What I can say is that these viruses are cutting in a place that the host has designed to cause like inflammation and have an immunological response. So it's tricking the virus into triggering the immune system.
Wait, so you're saying the host cell, so whatever cell the virus is getting into, has a sequence that looks like the big noodle protein, but it's like actually just a trigger in the real host cell.
It's like these viruses have brains, huh? You would think.
That's wild.
But why would you want to do that?
So, well, I guess the host is, that's why there's this evolutionary arms race, right? Like it's probably that a long time ago or in another species, like it would cut and it would actually do damage to the host or actually continue, like, benefit the virus. But now the host is like, nah, dude. Like,
I'm gonna get you.
Yeah.
You on my list.
But it seems that some viruses are still able to, like, evade this immune response.
Interesting.
And so I'm studying, like, how.
How do they know?
Yeah, how
I
they
love
know.
that.
How are these viral proteases, like, what else are they targeting, kind of thing. So I guess I think that's big enough. I don't know. I don't want to get in trouble.
You won't.
So these are the rotations we have been in. Rotation 2 ends December 18th, and then we have a couple of weeks off, and we'll come in January and start our third rotation. Whee! As we mentioned before, Asama and I will be in the same lab.
Yes.
I will be kind of co-advised by someone in engineering and someone in biology, which is super exciting. And that's exactly what I wanted to do. I was like kind of scared of getting into engineering things in undergrad because I was like, I don't have the background, I don't have the math, I don't have the physics, I don't have blah blah whatever. And then I come to UCSD and they're like, hey, we have this thing that's combining, trying to collab between the biology programs and the engineering departments. And I was like, wait, this is really cool. I'm super excited to do it. Everyone has been super nice recently, so hoping that turns out well.
I'm so excited for you.
Do you have to take classes from the engineering?
No. The thing is, they want us to be biologists.
Like, they want a different perspective.
Exactly.
Yeah.
Like, they want us to take everything we know from biology and be like, okay, this is what we're trying to do. And we'd be like, actually, you can't do that because cells need this, this, and that. Right?
Yeah, I think that's like an intersection. Like, science as a whole, or biology as a whole, I feel, has become really interdisciplinary. And I think that bridge between, like, life sciences to engineering is sometimes lacking. So I am excited for you to be stepping onto that bridge.
Thank you.
So yeah.
I'm so proud of my friends.
Oh, don't do that to me right now. Stop. Okay, so I feel like we're good on rotations. We feel fulfilled. I feel like is the
we're so lucky though, because
Yeah.
like we do have to caveat that, you know, sometimes people have to schedule their rotation for later semester due to like COVID restrictions, or now that a lot of people are working from home, they have other responsibilities or duties, so they might not feel like they could take a student right now. So we're really thankful for like all these opportunities, but we also like recognize that a lot of people are trying to make it work.
Yeah. I think we also recognize sometimes that labs don't pass the vibe check.
Yeah.
And that's okay too.
Yeah, some people have very different experiences in
Yeah.
their rotation, and that's fine. That's why you vibe check.
Exactly.
That's why we do this.
And sometimes your first couple weeks, you're not even sure if there's a vibe. 'Cause you barely see people. And then, you know, ask me again in 2 weeks. And then I'm like best friends with my lab manager. So,
yeah. And if you are listening to this and you're like, I don't know if I'll be in a lab or I'll be in a program that has rotations, you can definitely check online. A lot of places and like programs will have their requirements online that you can look through and see what's required of first year students. Do you have to come in under a direct admissions under a specific professor, or can you hop around like we are? So, and with that, let's talk a little bit about classes because
yeah, what else are we doing this semester? I mean, this quarter, sorry.
Yeah, it's okay. I come from a semester system
Me
too.
too.
It just comes out of my mouth so easy.
I don't think I'd ever. Yeah, no. Semesters? What are those?
16 weeks?
You just have 2 things.
Yeah.
An optional summer.
Oh, interesting.
So I'm taking 3 classes?
Yeah.
Yeah, you guys are taking 3 classes too. So Asama and Lennice are in a class that I am not in, and I am in a class that they are not in, which has been interesting.
But then we overlap in 2 classes. So
We do.
Yeah.
yeah.
Yeah, so I'm taking stem cell biology, or current concepts in stem cell biology.
Current, so not old.
No, it's like some newer papers, which I think is really cool because the postdoc I'm working in my rotation lab right now said he took the same class when he was a grad student here.
What?
And was it different?
because I think that there wasn't as much literature, or like there was different kinds of literature. Like they were still figuring things out. But like we have progressed so much in the stem cell field. There's still a lot to learn and a lot of growth to be done, but like so much has happened in the last like 3 or 4 years.
Wow.
So Amanda's in the stem cell class, and then me and Asama are in the bioinformatics class. And I think it's been really helpful. I don't know. I've heard there's mixed reviews, but I love it. At least I love the instructor. I think he's really, really helpful and really, really empathetic.
Yeah, And instead of, making us sit through like 4 hours of class and lab, like at very specific time, he recognizes that, it's hard for people to learn from home or even make time and try to figure out COVID schedule. So we have sort of like a hybrid schedule where he would post videos so we can watch them on our own, and also like try to work on a project. But also on Friday he has a YouTube live cast, which is always fun because you can like live chat the instructor, which is like a luxury these days. But last week
you're gonna talk about these kids.
Or this week, this is actually cute because it makes me feel like our professors are humans too, and they're dealing with like real problems. I guess he was trying to like draw something for us, like to explain a concept, but then he said that his kids mess up the iPad and then they were like trying to get into the office and they were like screaming, and then later we were like, oh, can you just like put them on the screen so we can say hi to them? And that happened, and it was really cute.
It was very cute.
Yeah, so I love that class.
They were like, hello, and he's like, okay.
Yeah.
Kids,
and also like things that I learned from that class actually helped me a lot during my rotation where I had to do bioinformatics. I felt like I was more confident about like figuring out my own data and how to work out my project. So it's really helpful.
Yeah, and I think the class in itself is really applied in that a lot of the concepts that we learn are for specific kind of techniques that may be used later on in our research. Like we did some RNA-seq analysis
that's fun.
And stuff like that. So it's things that we can maybe actually use eventually once we start getting into these like bigger data sets later on in our research. And then the two other classes that we have together are
core, which stands for concepts on research, experience, experiment.
I thought it just stands for core.
Yeah, no, it stands for something.
Concepts, something, reasoning? No.
Oh, maybe it is reasoning. I don't know. I just always call it
I'm
core.
checking Canvas.
I just always call it core. So we have core 1, and then we have core 2. Is there a core 3 if we're on a quarter system?
Oh no, I think there's just a core.
I think it's just
core 1,
core 1, core 2. So that's our core class.
She made a joke, ladies and gentlemen.
Thanks for recognizing that. And then we have basically, it's a how to be a grad student class on Tuesdays.
Yes.
And they call it pizza class because we used to get pizza. But now we don't.
Yes.
Someone sounds salty.
I am. Listen, everyone's like, every time I go somewhere, they're like, oh, usually we do free food for you guys, but you guys aren't going to get any.
Not during the pandemic.
Not during the pandemic. I'm at home at Zoom, and then it's like kind of weird to just eat on camera, but it's like I should be eating food right now.
Yeah.
So.
I was really looking forward to free food at school.
Yeah. It's bagels, bro. Carbs.
Yeah, maybe it will come back.
I looked it up. It's concepts of reasoning and experimentation.
Oh, wait, so I wasn't far off.
You were not.
I had reasoning.
Yeah, that's all I
was.
needed. That's all I needed. Teamwork.
But yeah, I'm kind of salty we don't get pizza, but it's okay, I guess.
Yeah.
One day.
I finished my last stem cell class this week, and they were like, oh, usually on the last day we bring pizza to treat you guys. I was like, why would you say that to me? Why would you say that to me?
Yeah.
Oh my god.
I'd rather have been ignorantly blissful that this is just the end of class, you know?
Yeah.
Like, if you're not going to give me a Grubhub gift card, whatever, don't mention it. Venmo me. Like,
my
Put
Cash
that
App
in
and
my
Venmo
student
are...
account.
Yeah, listen.
But yeah, that sucks. I guess the pizza part of the pizza class that go along with a lot of the like social things outside of class and lab that we don't get now that
Yeah.
we're doing a PhD in a pandemic. I just love saying that way.
Our choices.
I wonder how other first year PhD students are doing.
I have a friend
Their
that just
fall
started
semester.
their PhD at Notre Dame in physics.
Mm-hmm.
That's
So
a
I
different
think-
world.
That is
Yeah.
a very different world. But maybe we can ask him. He used to be on my dance team. So maybe I can send him a message and we can
Yeah.
talk to people.
Yeah. Maybe we should. I mean, I'm sure you know. I'm pointing at Asama since this is a podcast. Asama, you probably know other people who just started PhDs. I did too. Maybe that's a good little question we can all kind of.
Yeah, but do they have microphones?
But we can ask and just.
We're gonna have our Zoom audio. How do y'all feel
I
about
don't
that?
know.
Have them send like a voice message.
LOL.
And we put it right next to the microphone.
Call on FaceTime audio.
Yes.
We can also just have them comment and us like talk, be like so and so.
Yeah.
I don't know.
No, maybe we should have it live. It'd be fun. I miss that friend. So
Yeah.
it'd be good. They sent me a podcast I might like this week, which I thought was really cute.
And did you send our podcast
I know.
to
We like.
them
What about us?
in return?
Wait, I should have done that.
Yes.
I don't know if he listened or not.
You
Maybe I'll
missed
check.
out on the
What?
shameless plug.
Oh,
know.
I stay plugging our podcast. I be in Zoom chats like anyway, so there's this podcast on all streaming platforms. You can follow them at Triplicates Pod on Instagram and Twitter. You know.
I
Your
love
whole
that.
spiel.
A whole little thing. Yeah. Me and Asama were on a grad student panel for one of the organizations here at UCSD on Zoom, and I definitely said, "Oh, Asama's podcast is available on all streaming
Oh my
platforms."
God.
I didn't want to be like seemed so.
No, but then luckily, a few people follow you on Twitter, and
Oh,
they
so
were
they've
like,
seen it,
"This
yeah.
is Amanda's podcast too," so it's okay. I still can't get over like how supportive people are
Yeah.
of even like our first 16-minute
I
episode.
cried.
Yeah. Shout out to
yeah.
the UCSD Division of Biosciences.
Yes.
Like definitely all the faculty members have gone hard for us.
I know.
It's so nice to see.
Yeah. Also, what's it like to have faculty who know how to tweet? Like, that's a different world.
Some of these faculty have bigger followings than I do.
Wait, I want to know who.
What do you mean?
Whomst?
Whomst has more followers
we
than
can
me?
just say.
Fuck
Oh, I know.
yeah.
Okay. Oh, he does. I'll get there. I'm only like 300 behind him.
Oh,
I
okay.
am.
Yes, Amanda, use your fame to promote it.
Listen.
I think I tweeted about this. I was like, y'all, thank you for like supporting us because
Yeah.
it really shows me that UCSD was the place I wanted
Yeah.
to be.
Right. This is a wonderful place.
Yes.
So far.
Let's not put bad vibes into the atmosphere.
No, I'm not saying. I'm just saying we've been here for 10,
Yes.
14 weeks, however you want to start it.
I agree.
Yeah. So I don't want to be like,
Yeah.
whatever, and it'd be kind of weird, but like,
Um,
no, I love it here. I love San Diego. I send pictures of my friends of me at the beach. I'm like, hey, just a reminder, this could be you. But y'all playing.
That's how you recruit.
in December.
In December, I'm at the beach. I went to the beach today
Oh my
to
word.
finish my fellowship apps. Did I finish it? No.
Maybe for recruitment, we should just like tell applicants about all our beach
Listen, I am on the
experiences.
recruitment committee.
You know what you should do is have someone just sit at Zoom and just be like, oh, this isn't a virtual background. Like, we're just at the beach right now at the glider port.
But internet connection.
My iPad has cell service.
Oh
Like, just
wow, she said money.
Cut that out. We tried to get funded.
Yeah. No, but honestly, getting cell service on your iPad is the best decision. Because then you can do your work anywhere. And it's not like, oh, I need to be at a coffee shop because Wi-Fi. It's like, oh, I can read my papers at the beach.
Yeah, not that coffee shops are open right now.
Because they're not open right now.
And they shouldn't be.
Nope. But the beach is open.
Mm-hmm.
For better or for worse. I try to stay away from people.
Yeah.
Yes.
But the good thing is, like, there are so many beaches in
That's true.
San Diego. Like, not everyone is going
To the same one.
to, like, the
Mhm.
downtown one.
And
or-
generally, people respect your space.
I think it depends where you are. Because if you're, like, by the ocean, like, the ocean, there's people that, like, run up the coast.
Yeah.
And they usually aren't wearing a mask. And they'll just run right in between y'all. The surfers, too, will walk right by you if you're close to, like, wherever they come in and out. And they have no masks on. And they're obviously talking to all these other surfers.
Yep.
Yeah.
So
Listen, wear your mask. And let's all remember, it goes over your nose. It is not a chin strap. Thank you.
Not your earrings.
Not your earrings. That's a good one.
say Hoops 2020.
Stay safe out there, y'all.
Yeah. Okay, let's round out our episode. Let's do some observations. Let's run some stats on our data. What are we looking at this week?
Okay, so the question we're asking this week is, how many PhD students in our study have completed schoolwork at the beach? Amanda? Oh, Lennice can go. Sorry.
I do be reading papers on the beach, so yes, I have.
I also have gone to the beach to do some schoolwork.
Wow. I actually haven't because I'm not sure about getting sand on my keyboard. But now that you mentioned about the iPad, I might start doing that.
Yeah, I do worry about the iPad, though. Still with the sand, like the case. But listen, today I went to the beach and I stayed in my car because I was tired of these people with their chin-strapped masks.
Exactly.
And I just popped I have a little hatchback. I popped the hatchback, reversed into a parking spot, and just sat and looked at the water.
That's so smart.
Yeah.
It's basically a big chair.
It is a big chair.
Your car is a big chair.
There's a big chair on campus. Very big digression, but anyways.
What?
Wait, what big chair are you
There's
talking
like a big red chair.
What
Like
are
a
you talking
huge
about?
red chair.
Where?
Like an Adirondack
I don't
chair?
know, but it's on campus.
Have you seen it with your own eyes?
I've seen a photo of it with
Okay,
my own
so you
eyes.
haven't
So
seen it
doesn't
with
know
your
what
own
it
eyes.
is.
So
I
it
will
doesn't
find it.
exist.
Oh, okay.
But I have seen those things at like a pumpkin patch or like
No,
a
but
farm.
it's bigger. It's huge.
I've seen Fort Lauderdale had like a big chair in downtown and people would like climb in
Yeah,
it.
I've seen one in Boston too,
yeah.
actually.
Where you can
Like
see
near
multiple
Newbury.
people. Anyways.
Yeah. Anyways, big digression.
Our study.
Yes, our study. So 66.6666% of PhD students in our study have completed schoolwork at the beach. Again, you can find us on Twitter and Instagram at Triplicates Pod.
And if you have a story about your first quarter or semester that you would like to share with us, feel free to email us at triplicatespod@gmail.com and you might just hear it on the show.
And with that,
Obrigada. Ciao.
gracias y adiós.
Kabun ka. Bye.